to my dearest little champions. How are you all? I hope everyone who is watching this video is doing fine, is putting their 100% effort into studying all the subjects which is there. Now I know that a lot of you might be thinking that I don't know where to start from. There's a lot left. There is PU2, there is this, there is that. KCET is just like 1 or 2 months away. That's all. There's so less time. But let me tell you all, if you take the initial step, you never know when you're gonna reach there, the aim that you're looking forward for. For that, you have to take the step. Today you do 1%, tomorrow you do 2%, day after tomorrow 5%, and then 10% and probably 100 someday. So why not just try it out, and God knows, it might just work out for you. So I hope that you all are in a spirit now to study for all the subjects which are difficult for you, for all the subjects that is easy for you, and you're gonna score really great marks in KCET 2025. Now if you're in, do let me know that you're in. You're say that I'm gonna score full on full in biology in the comment section. If you think that you can, try. Okay, if you think that you are ready to try, comment it down. And I will start with molecular basis of inheritance in today's Masterstroke series, which I know is a very difficult chapter. Not very difficult, it's very easy chapter. Let's say it's easy, but it's kind of huge. Okay, it's kind of huge, which is why it seems difficult to us. But today we are gonna decode it really well, and we are gonna score what? Full on full. Actually, you are not saying full on full. I am only the one who is saying full on full. Chalo, first thing. The DNA structure or the way RNA structure, whatever genetic material you take, DNA, RNA, the basic unit is what? A nucleotide. What do you call it? Nucleotide. And this is going to be really fast, okay? This is a one short session, very fast. So nucleotide contains 3 components. One is the nitrogenous base, second is the pentose sugar that you are having, and one is the phosphate group that you have. All of them come together to form what? Basically the nucleotide, my dear children. If we talk about the nitrogen bases, you have 2 types. One is purines, another one is pyrimidines. Purines are adenosine and guanosine, or basically adenine and guanine, not adenosine and guanosine, adenine and guanine. And if we talk about pyrimidines, it is cytosine, thymine, or uracil. So thymine is there in DNA, but in the case of RNA you have uracil. That becomes a major difference between DNA and RNA. Now if you talk about the pentose sugar, you have 2 types. One is deoxyribose, that is present in the DNA, and one is ribose, that is present in the RNA. What is the difference between deoxyribose and ribose? Basically, in the case of ribose, there is an OH over here. This is the first carbon of the pentose sugar, this is the second carbon of the pentose sugar, this is the third carbon, this is the fourth carbon, and this carbon that you see is the 5th carbon of this particular sugar which is coming outside. Now on the second carbon there is an OH group in the case of what? Ribose sugar. But in the case of deoxyribose, do we have an OH group? No. Cool. So these are the 3 components. Now formation of DNA. So it's not like the nucleotide comes as in together like that. There is step-by-step procedure. First thing, you just have the sugar and you have nitrogen base. Both of them come together. When there's just a sugar and the nitrogen base coming together, you call this structure as a nucleoside. Not tide, it is nucleoside, my dear children. So over here, the sugar, the first carbon of the sugar combines with the nitrogen base, and this bond which is being formed over here, it is known as none other than N-glycosidic bond. Now, the adenine which was there, you will call it adenosine now, or basically in the case of DNA, you will call it deoxyadenosine. The guanine which was there, you will call it guanosine or deoxyguanosine. Okay, the cytosine which was there, you call it cytidine. Okay, or probably deoxycytidine in the case of DNA. Okay, same thing goes for all the things, and this is what, my dear children, this is the nucleoside which is being formed over here. Talking about the nucleotide formation, what is nucleotide? I told you, you need the phosphate group also over here. So that bond also has to be made. Where do they make that bond? On the 5th carbon. On the 5th carbon, this will attach to the phosphate group, which is right over here, my dear children. And this bond is known as ester bond. This bond is known as ester bond, and since phosphate is also involved over here, you will call this as phosphoester bond. So first carbon with nitrogen base, 5th carbon with what? With basically your phosphate group, and this is known as phosphoester bond. That's how you make one nucleotide. But what if you want to make multiple nucleotides coming together? That is a polynucleotide chain, that is our DNA. How do we exactly make it? Let's see that. So nucleotides link via 3,5-phosphodiester bonds. Yes, so we were talking about that. Yeah, so this bond is known as, okay, now as I was saying, we need to make together nucleotides come together, right? So what do we do for that? Over here we have a third carbon of the pentose sugar. This third carbon also attached with the phosphate group. This is again an ester bond. This is again, my dear children, an ester bond. So 2 ester bonds and 1 phosphate over here. So what do you call it? Phosphodiester bond that is being made between the polynucleotide chain, my dear children. And now when you go ahead, okay, what do you see? Ayo, yes. When you go ahead, what do you see? Over here, the 5th carbon has a phosphate group. It is attached to the phosphate group, but do not have any attachment to the another sugar which is there, because there is no sugar up there. So this 5' end is free. So what do you call this end as? 5' end of the DNA. Similarly, if you go ahead over here, the 3' is not attached to another phosphate group which is attached to another, pentose sugar. No, nothing like that. So this 3' end is free. So I will call it as 3' end. Okay, so in the case of DNA, we say there is a 5' end. We say there is a 5' end and there is a 3' end which goes like this. Okay. Now over here, RNA has an additional, backbone. So formed by sugar phosphate units, you saw that sugar and the phosphate comes together, that forms the backbone. Please remember that, with nitrogenous bases projecting outward. It's not outward, it's inward. The nitrogen base which is there, it always projects inward. So basically, if suppose this is a DNA, the nitrogen base is coming like this. Similarly, there will be another strand of DNA where the nitrogen bases will be coming over here. So nitrogen bases will be bonding with another nitrogen bases which are inside, they are sandwiched. Okay, please remember that. Now RNA versus DNA, RNA has an additional OH, I told you, at the second end, 2' end of the 2' carbon of the ribose. RNA contains uracil instead of thymine. So it's not 5-methyl uracil is for thymine, okay. The other name for thymine is 5-methyl uracil. So RNA contains uracil instead of thymine, or what do you call it as? 5-methyl uracil. Please don't say uracil means this, okay. It's about thymine that you're talking about. Got it? Chalo. Next thing. Now, the DNA double helix model, how it is actually made. The DNA, it is coiled like this in a double helical structure. But what are the salient features of this double helix model? First thing, 2 anti-parallel polynucleotide chains with a sugar phosphate backbone and bases projecting inward are present. So over here, if I say that this is this particular DNA which is going, this strand which is going over here, my dear children, this is from 5' to 3' end. The other one will be going anti-parallel, and this will be in 3' to 5' end. Cool. These are 2 anti-parallel polynucleotide chains that you are having. Now, base pairing via hydrogen bonds, that is also there. So I told you the nitrogen bases which are there, they are inside, they are sandwiched. They are kind of together. They have bonds between them. What are these bonds? They are hydrogen bonds, my dear children. These are what? Hydrogen bonds. So basically, if I say I have, suppose, guanine over here. Guanine will be forming 3 hydrogen bonds with cytosine. What about adenine? Adenine will be forming 2 hydrogen bonds with thymine. This is the case of DNA, my dear children. So how do you remember 3 or 2 hydrogen bonds? Basically, G and C looks more similar, so they are tightly bonded. You just think like that. So 3 of them are there between G and C, and 2 of them are between A and T. Now, if it was an RNA, my dear children, A will be binding with U, not T. A will be binding with U because in RNA there is no T. There is no thymine. Cool. Now next thing, right-handed helix. Okay, there is a right-handed helix which is there, and the pitch of 3.4 nanometer. What do you mean by pitch of 3.4 nanometer? This is a pitch where you get one whole helix over here, my dear children. This whole thing will be counted as a pitch. And what is it, my dear children? Okay. What is this? Sorry, I will count this whole thing as a helix, and the pitch of it will be 3.4 nanometer. In this particular helix, you will be finding 10 base pairs. There will be 10 things like this. These are known as base pairs. Okay, how many? 10. Now, I am telling you one more thing. The space between this base pairs, my dear children, is 0.34 nanometer. Is what? 0.34 nanometer. Cool? So, right-handed helix with a pitch of 3.4 nanometer, 10 base pairs per turn, okay, and 0.34 nanometer per base pair in between these base pairs. Now, base pairing, stacking and H bonds provide structural stability to the whole DNA double helix. Got it? Please remember this whole thing. Helix 3.1 pitch, one whole helix has a pitch of 3.4 nanometer. In between each base pairs, what is the space? 0.34 nanometer. And how many bases are there? 10 bases. Now, discoveries that you should be knowing. In 1869, my dear children, Friedrich Mischer was the first person who said that. Basically, he is saying some acidic substance in the cell nuclei and he called it nuclein. What is this acidic substance which was there? It was actually DNA. DNA is acidic in nature. It's negatively charged. Okay. What year? 1869. What is the name of the guy? Friedrich Mischer. He is a Swiss physician, very much respected person. Okay, he found an acidic substance in the cell nuclei and he called it nuclein, my dear children. Now, in the year 1953, long after that, my dear children, James Watson and Francis Crick proposed the DNA double helix model, utilizing what information? Utilizing the information from S-ray diffraction technique done by Maurice Wilkins and Rosalind Franklin. So, these two people were doing the S-ray diffraction technique by which they found out some data, and on the basis of this data, James Watson and Francis Crick in the year 1953 gave the double helix model. Okay, now who spoke about the base pairing? Their model incorporated Erwin Chargaff's findings that in double-stranded DNA, the ratios of adenine to thymine and guanine to cytosine are constant and equal to 1. So basically, how much ever adenine is there, that much thymine will be there. How much ever guanine is there, that much cytosine will also be there. That is what was said by what? Erwin Chargaff, and this is known as Chargaff's rule as well. Okay, please remember it. Cool? Chalo. So, please remember 1953 and 1869. Now, what is the dimension of a nucleus? If you talk about a nucleus, what is the dimension of it? It's just 10 to the power minus 6 meters. 10 to the power minus 6, that many zeros are there in front, okay? You see how small it is. But what is the length of our DNA, my dear children? The length of our DNA, how many base pairs do we have? We have 6.6 into 10 to the power 6 base pairs. How much? 6.6 into 10 to the power 6 base pairs we have, and in between each base pair I told you that there is a space of 0.34 nanometer. Okay, 0.34 nanometer, which I take in meters, it becomes 10 to the power minus 9 meters. So, total thing comes out to be 2.2 meters. So, 2.2 meter thing, how can it be fit inside 10 to the power minus 6 meter nucleus? For that, you have to do something. So, how is this long DNA put inside the nucleus, my dear children? By wrapping it around some proteins which are known as histone proteins. What do you call it? Histone proteins, my dear children. Okay, so initially, what did we talk about? We spoke about the nucleotide. Okay, we spoke about nucleoside, then nucleotide, then how polynucleotide thing was formed. Then we spoke about the common discoveries which were there. Then we spoke about the nucleus. What is the size of the nucleus? Now we are trying to fit in this DNA that we discussed into the nucleus. How, my dear children? With the help of the histone proteins which are there. Okay, so let's see. What is the structure? So over here, this whole thing that we see is known as a nucleosome. What do you call it? It is known as a nucleosome. And nucleosome basically consists of histone protein. How many histone protein? 8 histone protein. And what do you call it? Histone octamer. What do you call this as? Histone octamer, because it is made up of 8 histone proteins. Okay. Now this histone proteins are made up of amino acid, right? Suppose the histone proteins has more amount of positively charged amino acids such as lysine as well as arginine. Then what will happen? The negatively charged DNA can easily wrap around it. That is the use of it. We want to make it complex, right? We want to make it compact, not complex. We want to make it compact. So that what we are doing, we are taking the histone proteins, we are wrapping around the DNA so that it becomes restricted to one particular location. That is, it can fit into 10 to the power minus 6 meter nucleus. Get it? So for that, what do we have? 8 histone proteins, which is known as histone octamer, my dear children, and the amino acids which are there, it is arginine and lysine, very important thing. Now the DNA will wrap around twice, only twice, and how many base pairs will be covered? Almost up to 200 base pairs will be covered over here. Now, this is the histone octamer, this is the DNA. Now, you have another histone protein which is known as H1 histone protein. Now, again, you will be having another nucleosome and the thing will be going on. So, nucleosome is basically a combination of the DNA as well as the histone protein which is there. Together, you call this as nucleosome, my dear children. What is this particular protein that is there? H1 histone protein. Okay. Suppose I am saying that and this structure, okay, suppose so many of them are together. So many of them are together. What do you call this whole structure as? If you see, they look like bead on a string like structure. This bead on a string like structure is known as chromatin. It is known as chromatin, my dear children. Suppose these beads are very, very close to each other. That means in between two beads, their H1 protein will be also very close. So this scenario is given to you that basically the H1 protein is kind of very close to the nucleosomes. What is happening over here? What type of chromatin is being made over here? So chromatin are also of two different types. We are going to talk about that. So over here, my dear children, as I said, the nucleosome units comes together to give a bead on string like structure called as chromatin. There are certain non-histone proteins also. Okay, it's not just histone proteins will be there. There will be some non-histone chromosomal proteins, that is NHC, that will be required in the chromatin. Now, the chromatin is of two types that I was talking about. One is euchromatin, another one is what? Heterochromatin. Euchromatin is loosely packed. That means the beads are kind of far away from each other, and there will be one H1 histone protein in the middle. So they are loosely packed and they stain really light. It stains what? Really light, my dear children. And I'll call this as transcriptionally active chromatin. What do you mean by that, ma'am? Basically, why do we need DNA for making proteins, right? So this information which is there in the DNA, it's not understood by the people who make the proteins. So basically, you transcribe this information into some other language and you call the structure as mRNA. So from the DNA, you transcribe the information into mRNA, my dear children. This process is known as transcription. And for that, you need enzymes and all who can read the language. So when I say this is euchromatin, I say that it is transcriptionally active. That means this process can be easily taking place over here. So euchromatin is loosely packed, it stains light, okay, and it is transcriptionally very active. What about heterochromatin? It is more densely packed and stains really dark, and it is transcriptionally inactive chromatin, my dear children. Okay, chalo. Next thing. The search for genetic material. So until now, okay, so now one problem is sorted, that is we can fit our DNA into the nucleus by converting it into a chromatin, okay, into a more condensed way. Now the chromatin, when it gets more condensed, what do you call it? You call this as a chromosome. Okay, when the chromatin basically gets more condensed, you will call this structure as what? A chromosome, my dear children. Okay, now that was there. Now parallelly, one major thing was also happening. Okay, parallelly one major thing was also happening, but before that I want you to take, okay, 1953, okay. So over here, other people, okay, in 1953, your James Watson and Francis Crick found out the DNA double helix structure and things like that. That is okay. In 1928, much more before that, there were certain people who were thinking like, what could be actually the genetic material? So they were in their search of the genetic material, and for that we have three experiments. First one is the transforming principle experiment, my dear children, which was conducted by Frederick Griffith in the year 1928. For that, he used a bacteria that is Streptococcus pneumoniae, as you can see over here. Okay, and there are two strains of the bacteria. One is S strain. Okay, another one is R strain. Why do you call it S strain? Because basically the S strain have a mucus polysaccharide coat, making it virulent. So it was very smooth because of the extra polysaccharide coat which was there. But this was also making it virulent, my dear children, as in it was kind of toxic. If some animal or basically some organism get affected by this particular bacteria, there are high chances that they will be dead. Okay, now talking about R strain, it did not have that particular coat and hence it was non-virulent, my dear children. So how was this particular information being actually used? So Griffith's transforming principle says that he took a live R strain, that is a strain which was alive, that is a bacterial strain, okay, the R strain which was non-virulent. He put it into a mouse. He saw that the mouse basically it lived, it didn't die. He took a live S strain and he put it into mouse, and he found that the mouse basically became dead, okay, because it was the virulent form. Then he took the heat killed S strain. He treated with heat and basically killed the S strain, and then he put it into what? Basically the mouse which was there, and he found that the mouse lived. That was kind of strange. He just heated it. How can this particular guy live? This strain is supposed to be kind of very toxic. It should be causing the disease, right? But there was something that was not common that he saw. Then what he did is what? He took heat killed S strain, okay, heat killed S strain which was used over here, and he took live R strain. Live R strain never caused any trouble, but when it was mixed with this, the mouse still died. Both of them actually, when they were separate, they didn't cause any trouble. But now over here, the mouse is dying. How is that possible? Then he took live R strain from group 4. Again, basically, the mouse, this is not needed. At least you study until here. Okay? So, he tried to understand this. He thought that something probably is getting transferred from here to here. Something is there that is not getting damaged by heat, and it is still getting transferred from here to here and making this live R strain as convert into a live S strain. This converting the live R strain into live S strain, how? Because something is getting transferred from here to here. So, some transforming principle transformed from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. Got it? So, he was unsure about what it could be, whether it's RNA, DNA, protein, what. So, then came other people, Oswald Avery, Colin MacLeod and Macklin McCarthy in the year 1933 and 44. So, they did some experiment, okay. They took the heat killed S cells, which was not causing any trouble anyways, okay, and then they added proteinase, okay. They added what? Proteinase. So, basically what does proteinase do? It kills, basically damages all the protein. So, the protein was not there. From in the heat killed S strain, whatever protein was there, it has been lost now. And now, this heat killed S strain was mixed with R strain, the live R strain. This time he found that basically nothing happened. Okay, the mouse was still dying. The mouse was still dying. When this whole mixture of thing was being put into the mouse, it was still dying. That means transformation was still taking place. Then again he took something and put RNase. He took the heat killed S strain again, put RNase, treated with RNase, that is, it damages the RNA out there, and then mixed with R strain. Again he found that no transformation is happening. Basically the mouse is still dying. There is no effect. Okay. Then he took one thing. Again heat killed S strain, and he put DNase, that is going to damage the DNA of the heat killed S strain if at all it's there. And then he mixed with R strain, and then he found that basically the mouse didn't die. Sorry, the mouse didn't die. Yeah, the mouse didn't die. Why the mouse didn't die, my dear children? Because transformation was not taking place. The DNA which was there, probably it might have been getting transferred. If I am treating with DNase, obviously this DNA is going to get damaged, there is no transfer going to happen, and hence there is no conversion, there is no formation of S strain that is going to take place over here. Initially the R strains were getting transformed to S strain, which is why all of them were dying, but over here there is no transformation that was appearing. So, no S strain, no S cell appear over here, my dear children. With that, they conclude that probably it's the DNA that is the genetic material. So, method is what? Isolated components, proteins, RNA, DNA from the heat killed S strain bacteria. Introduce each component to live R strain bacteria. Okay. Protease and RNase treatments did not prevent transformation. DNase treatment inhibited what? Transformation. So, either you do what? Basically take all the proteins, everything together and introduce each component to the R strain, and along with that you add this one by one these things. You will get to know what is it. So, exactly this was done. It's written in a different language over there, but means the same thing. If it's easy to follow this, just follow this, that's all. But still, it was not kind of, they were not kind of very sure. Even though they were sure, people were not sure. So, one more experiment was needed, and this was the last experiment for the genetic material. And it was done by Alfred Hershey and Martha Chase, my dear children, in the year 1952. And 1953, what happened? Basically, James Watson and Francis Crick did the double helix model of the DNA. So, over here what was done is that they took two bacteriophage. Just a second, okay. They took two bacteriophage. This is the bacteriophage. Okay. Bacteriophage has its own genetic material. Now, they labeled this bacteriophage with S35, sulfur 35 isotope. Now S35 becomes a part of the protein which is there, protein capsule which covers the whole bacteriophage, because sulfur is a part of it. Okay. Then one more bacteriophage was taken where it was labeled with radioactive P32. Now P32 is a component, okay, basically phosphate is a component of the DNA, so it went and DNA, or basically the genetic material, whatever is there, RNA, DNA, whatever is there. So it went and get accumulated or basically incorporated into the genetic material over there. So P32 with the genetic material and S35 with the protein coat which is there. Now he made this bacteriophage go and infect an E. coli bacteria. Okay, now what happens is that usually the bacteriophage will be putting its genetic material inside the host. So it did it, okay, and the capsule which was there, it was left behind outside. Now what was done, basically this whole thing, okay, was made to sediment, and he found out that basically in the cells there was no radioactivity. In the cells there was no radioactivity to be seen. That means the protein is not anyways getting transferred, because the protein is the one that is having the radioactiveness. So it is not the protein that is getting transformed. Next thing, he saw that the supernatant which was there, okay, they saw the supernatant which was there, it was containing radioactivity. That means the supernatant that contains the proteins were left outside. It was not something that was passed from one organism to another organism. So they got to understand that. And the opposite thing happened over here. Same thing, infected, it passed its genetic material. Now what happened, this radioactive genetic material is within the cell. So obviously the cells would be radioactive now. And what makes the cell radioactive, my dear children? The genetic material which was labeled. So it was found that basically it's the genetic material that is getting transformed. And what is it, my dear children? It's the DNA that is getting transformed. Okay. It is the DNA that was getting transformed. No radioactivity was detected in the supernatant, the thing that was left behind. Get it? So this was the whole experiment, and with this it was cleared that basically DNA is the genetic material. Got it? So 3 experiments, you have to remember the name, remember bacteriophage, remember E. coli, their Streptococcus pneumoniae, all of them. Very much important. Going ahead. That is done. So first genetic material, basically the genetic material is DNA that was being identified. But what was actually the first genetic material when we talk about evolution? It was none other than RNA, my dear children. So was it only acting as genetic material or anything else as well? Well, yes, it was also acting like enzyme, a catalyst. Now what is the problem with RNA being both an enzyme as well as a genetic material? The problem is that when you say that one particular molecule is a catalyst or an enzyme or doing things like that, basically it will be highly reactive. It will go react, change its structure, change something else's structure. So it will be highly unstable. So can you have something in your body which is highly unstable as a genetic material? This genetic material is basically the one that will be coding for each and every protein in your body, my dear children. And if that is something unstable, can you rely on it? No. Which is why the RNA which was there, it was evolved into a much stable form which was DNA later on. Okay. Now this is a small comparison chart between the DNA and RNA that you should be definitely knowing. So why I say that DNA is better? Let's see. Sugar, first thing, deoxyribose, it lacks one oxygen which is there. I told you in the ribose sugar there is OH group, right? So here they have an extra hydroxyl group, but here they do not have it. And this hydroxyl group, my dear children, makes this particular RNA. If you ask me why RNA is reactive, it's because of this particular hydroxyl group which is there. Talking about the DNA strand, mostly it is double-stranded. Okay, but if we talk about RNA, it can be single-stranded and also double-stranded as well, but mostly it is single-stranded. Nitrogen bases, it has thymine. And here it has uracil that replaces thymine, my dear children. Thymine is what? 5-methyl-uracil, and it is again something that makes the DNA much more stable. Function, mainly it acts as a genetic material, that is DNA. What about RNA? RNA, you know, I told you, RNA, basically the DNA's information which is there, it has to transcribe into mRNA. This mRNA will be able to make proteins later on. So basically, can I say that this thing, RNA which is there, it is involved in protein synthesis? Yes. And also in transmission of information. In transmission of information, you more commonly use what? RNA, my dear children. Now, types. There is only mainly one type. Okay. You have multiple types of RNA. mRNA, that is messenger RNA. tRNA, okay, that is transfer RNA. rRNA, that is ribosomal RNA. rRNA makes the ribosome. tRNA is the one that will pick amino acids and say that, hey, amino acid come here, hey, amino acid come here. And all the amino acids are grouped together to form protein. Okay, it helps the mRNA to make the proteins. Now, location. DNA is mainly inside the nucleus and the mitochondria, not in the cytoplasm. In the cytoplasm, it is highly dangerous. There is lots of enzymes up there. What about the RNA? RNA is found in the nucleus as well as the cytoplasm, my dear children. Okay, so the DNA which is there in our nucleus, I told it transcribes the information into mRNA. DNA obviously cannot move out. So basically this mRNA is the one that will be moving out. So don't you think you would obviously have RNA in the cytoplasm? Yes. And I told you this mRNA is helped by tRNA. So obviously another form of RNA is also there. So it's all sorted. Okay, what about the stability? It is more stable and resists degradation. Okay, DNA. What about RNA? It is less stable and degrades really quick. Cool? So next, criteria of genetic material. So what I told you all the features. Now you tell me, okay, I told you the difference between RNA and DNA, and I am going to tell you about the criteria of a perfect genetic material. Now you tell me what is the perfect genetic material. Okay, so the ideal genetic material should be able to replicate, capable of self-duplication to ensure genetic continuity. So do you think DNA replicate itself? Yes. Does RNA replicate itself? Yes. Next thing, stability. Chemically and structurally stable to maintain integrity across an organism's life cycle. But DNA is more stable or RNA is more stable, my dear children? It is DNA which is more stable. With the thymine group, with no OH in the let's say second carbon, right? So it makes it more stable. Mutation potential, so allows for gradual changes to facilitate evolution. So it's not like the DNA should not be undergoing any changes, or RNA should not be undergoing any changes. No, there should be some changes in them, because these changes, my dear children, would lead to evolution. So you need mutation potential, but this should be less. It should not be like very rapid changes are happening. If there is very rapid changes, that will be a big problem. There should be small changes that should be happening. So again, who? DNA or RNA? DNA would allow minimal mutation to take place, but RNA, lots of it. Expression, able to manifest traits in accordance with the Mendelian inheritance. So whatever Mendel said, according to that, thing should be inherited. So again follows that it is the DNA that follows all these features, my dear children. Okay, now who is better genetic material? I told you it's DNA, but let's see. Is it actually DNA or not? So DNA is more stable due to complementary strands rejoining after heating. So basically this is the strand. If at all we separate it, still they will come together back. Okay, and this becomes more stable. Lack of reactive to OH, okay, in group that is there, and presence of thymine instead of uracil. All these reasons are why DNA is more stable. About mutation and evolution, my dear children. RNA mutates faster due to instability, and RNA-based viruses evolved rapidly. What about DNA? It does not mutate fastly, but mutation is definitely there. So basically, whatever points are there here, you're comparing it. Okay, so mutation is seen over here. It is faster due to instability, it is said. But in the case of DNA, it is also mutating, but not very faster. It's in a slow rate. Expression of genetic information. RNA directly codes for protein, whereas DNA relies on RNA for protein synthesis. That is there. In the case of expression of genetic, I told you the DNA's language cannot be understood easily, so it has to transcribe into mRNA. That's how things work. But if you talk about an RNA can directly form a protein, whereas in the case of a DNA, it relies on RNA, but it's okay. One particular feature, one drawback is totally fine. It's not a problem. Conclusion is what? DNA is preferred for genetic information storage due to its stability. That is all the more important, while RNA is better for transmission, as in information transmission or anything. Get it? Chalo. Now, DNA replication. How does this particular process exactly happen? Let's see. So, after the completion of replication, each of the DNA molecule would have one parental and one newly synthesized strand. So, if you see over here, this is a DNA strand. The DNA strand will unwind from itself, and there's a new strand that comes in over here. So, this strand which is already there, this is known as the parental strand. What is it? this is the parental strand, and this older strand which is there, okay, this new strand which is there, that is the newly synthesized strand. So whatever DNA you are going to make, you will find that one strand will always be parental, and this scheme was known as semi-conservative DNA replication. Okay, so you're not conserving the whole information which is there, but you are conserving at least one part of the information from the DNA of the parent. So that is what is known as semi-conservative mode of DNA replication, my dear children, again given by Watson. Now again, you need proofs in biology, otherwise how can you believe it? So there's one experiment done, okay, by Matthew Michelson and Franklin Stahl, and to prove the semi-conservative mode of replication, and this was done in the year 1958, much after Watson and Crick gave the DNA double helix model and stuff like that. So what did they exactly do? Basically, if you see, he grew the E. coli in a medium that was containing N15 isotope. Okay, now this N15 got incorporated into DNA, so the DNA double-stranded have what? Strands have what? N15 incorporated into them. Now if you try to sediment it, they will sediment in this particular range, my dear children, that is heavy, it is quite heavy. N15 isotope is really heavy. So both the strands are there, so I'll say it is going to sediment down up there like that. Now, what did he do is that he tried to grow the same organism in N14 containing medium, N14 containing medium. Now what happens? This DNA will separate and there's a new strand that will be formed. So the one that was already there, it will have N15, but the new strands which are going to form, obviously it will take over the N14 which is there in the medium. So now you will get two strands like this, which has one strand that is N15, another strand that is N14, my dear children. Now if you centrifuge it, you will find that it's not heavy. Basically it's settling somewhere over here, that is it is forming hybrid, my dear children. So this was done after 20 minutes. You know E. coli divides within 20 minutes. So after 20 minutes when he checked that is skim. Okay. Now again 20 more minutes, basically making a total of 40 minutes. What happened? Same in N14 medium only. What was happening? This will divide into two. As you can see over here, this will divide into two. A new strand forms, all of them N14 will be the strand that is found. Again what he found? He found that, okay, two of them they were hybrids and one was coming in the light portion, that is this, and this one was coming in the light region. So what do you conclude from this? This was happening, you could see these different patterns. Why, my dear children? Because only one parental strand is being conserved and there is another strand that is being added up over here. You get it? You get it? And this is how they said that DNA basically has one parental strand and there's a new strand being added, and this proved the semi-conservative mode of DNA replication. But what do you think that this is the only proof that you have? No, this is not the only proof that they had. They had one more proof. Taylor and colleagues in 1958, in the same year, my dear children, they used radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome, which was performed on Vishya faba. Please remember the name of this. What is it, my dear children? Vishya faba beans. So who? Taylor and colleague, they used radioactive thymidine for doing another experiment on Vishya faba, that is faba bean. Again my hair. Okay, cool. Will you be able to remember it? You have to remember it, and I know you will be remembering it. Okay, here E. coli was used. Now if you are asked what happens after 60 minutes, what happens after 60 minutes? So anyway, in all the generation you will find two hybrids. Okay, you will be finding two hybrids, that is for sure. Because the parental strand, the red one will always be there. Two of them were there from the start, it will be there. Okay, rest all will be green. Cool? Chalo. Which enzyme does DNA replication? Since we are talking about DNA replication, we should know the enzyme as well, right? So it is DNA dependent DNA polymerase. It's DNA polymerase which is totally dependent on the DNA strand for replication to happen. So suppose this is a DNA, another enzyme that is DNA dependent DNA polymerase will come and attach and make new copy of it. Okay? Now efficiency, what is the efficiency of this? So please remember this very important point, my dear children. E. coli, which has a total base pairs of 4.6 in 10 to the power 6 base pairs. Okay, it has 4.6 in 10 to the power 6 base pairs. We have 6.6 in 10 to the power 9 base pairs. Remember that. Completes replication in 18 minutes. In what? 18 minutes. With a polymerization rate of 2000 base pair per second. Now you can be asked, what is the polymerization rate of DNA polymerase in prokaryotes? E. coli is a bacteria, it's a prokaryote, my dear children. The polymerization rate is 2000 base pairs per second, and this question has come. Now, DNA polymerization from 5' to 3'N. So, when the DNA polymerization happens, my dear children, this particular enzyme which is there, it will be polymerizing from 5' to 3'N. That means, suppose this is 2 strand that we have. One is in 3' 5' direction, another one is in 5' 3' direction. So, over here the new strand, whenever it is formed, it is going to be formed in 5' to 3' direction. Okay. Now, DNA polymerization requires the origin of replication. So, DNA polymerase would not come and attach if the DNA strand which is there, it does not have an ORI sequence or origin of replication sequence. Please remember it. And one more thing, the polymerase reaction, which is the polymerase work that it does, okay, the DNA polymerase, it is highly accurate. It does it with high precision, my dear children, so that there is no mutation that is happening up over here. But still, there could be some errors, but very rare. Now, what is the energy source for this? So, I told you, basically DNA polymerase, it makes the DNA strand. How can it make the DNA strand? By taking nucleotides, by picking what? Nucleotides. There is one good thing about nucleotides. So, deoxyribose nucleoside triphosphates, my dear children. Why do you call it triphosphate? Basically, the thing is that when I say that this is nitrogen base and this is phosphate group, there are actually 3 phosphates in there. You break this bond, and that's how. You break this bond and then you attach this with another nucleotide. As of now, it will be known as triphosphate. It is known as triphosphate. So, deoxyribose, whatever nucleoside is there, my dear children, triphosphate are being used. So, each time when these guys are picked up, this bond is broken down, and when you break this bond, there is a lot of energy being released, and this energy helps in the formation of another bonds which are there. So, energy source is deoxyribose nucleoside triphosphate as both substrates, as in it is going to act as a substrate over here with which we can make it, and also as an energy source. Got it? Done? Chalo. Next. Steps of DNA replication. What are the steps of DNA replication? First, you have initiation, then you have elongation, and then you have termination. Very, 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 very easy. First thing, you know, DNA is there. Yes, DNA is there. You have to open this DNA, and for that, you know, there are hydrogen bases over here. Guanine, cytosine, you have 3, adenine, thymine, you have 2. So, DNA helicase is an enzyme that will come and unwind. It will open up the DNA helix at the origin of replication. From the origin of replication, this will be starting. Now, slowly, when they are separated, obviously this won't be stable. So, you need some proteins to attach. So, you call the single stranded binding proteins. So, these proteins comes and attaches over here so that they do not keep, they do not rebound to each other. Now, you might think it's open now. Suppose this was the case, and now I am going to open it like this. Now, it is being opened. It is being cut like this with the help of DNA helicase. I put single stranded binding protein so that they do not come and attach to each other. I started from origin of replication, my dear children. Now, you might think I will directly use what? DNA polymerase? No. Let me tell you one very important thing. Okay, this is suppose 3' and this is 5'. Okay, sorry, this is 5' and this is 5' and this will be 3'. So, DNA polymerases will be making strand from here, but it cannot directly do that. It needs pre-existing nucleotides up over here. And how do you get it? You get it by adding some primers over here. So, primers are a group of nucleotides which are there, and you are going to add it with the help of an enzyme, that is primase. The primase which is there, when it makes up the primer over here, that's when the DNA polymerase will be synthesizing it in 5' to 3' direction. Okay, cool. So, here it is fine. Here it is fine. But what about this strand? Here it is 3' to 5'. So, DNA polymerase basically will be working this side. Okay, but this guy will be opening up also every time. So, suppose I am adding a primer up over here. But still when I am okay opening, what will happen? Anyway, synthesis will be taking in this direction, right? Again I have to add another primer which will be making the DNA polymerase go from here to here. So there are so many primers being added up over here, and the DNA is also being added up like this. DNA is being elongated with so many gaps over here. What do you call this as? You call this as Okazaki fragments. You call this as Okazaki fragments. So over here where DNA is easily getting replicated, you call this strand as leading strand. And the other strand where it is going a bit slow because of the primers being attached and also an uneven thing. So you call this strand as lagging strand. What do you call it? Lagging strand. So let's see. So first thing, DNA helicase unwinds the double helix at the origin of replication. Single stranded binding proteins will come and attach to make the strands really apart. Primase acts what? RNA primers to start the replication. Now elongation process happens. What is elongation? So DNA polymerase add new nucleotides in 5' to 3' direction, as you can see over here. In leading strand it is made continuously. In lagging strand you have Okazaki fragments. Okay, there is not continuous DNA fragment formation over here. Lastly, what happens? Lastly, these primers, whatever are there, they are removed and the DNA are being joined with the help of another enzyme that is known as DNA ligase. Later on, after you're done with this, you do termination. How do you termination? Basically, suppose one DNA is getting open like this, another portion of the DNA is getting open like this. So two replication forms come and dash over here at one point. That's when the termination can stop, or can be some other mechanism as well. One thing I forgot to say, this thing that is being formed over here, which looks like a fork, my dear children, this is known as replication fork, where the DNA is being replicated. Now there is one more question that you should be knowing, that is, why cannot the DNA be totally unwind? Why cannot the DNA be totally unwind? Why there's a replication formation over there? Because the reason being that it is obviously going to take too much amount of energy if you want to open up the whole DNA. That is not at all possible. Okay, just a second. Now, where is it? Yes, components of DNA replication. What is it exactly? So unit origin of replication, I told you that, okay, a specific DNA region where replication will begin. Second thing, DNA helicase, which will be unwinding the DNA. DNA dependent DNA polymerase, which will actually replicate the DNA, and then replication fork, which is the small opening. DNA ligase, which is there, which is going to join the discontinuous fragments in the lagging strand. Okay, and also the leading strand, the primers, after it's been removed, you need to enlarge, but it's okay. Now DNA replication occurs in the S phase of the cell division. You know, in the cell division you have different cycles, right? S phase there is there, M phase it is there. Okay, so S phase is basically the synthetic phase, or you call it synthesis phase, where the DNA is getting replicated. Okay, M phase is where the DNA is actually getting, the cell is actually getting divided. Works? Chalo. And this S phase is a part of interphase. Okay. Now, how does DNA help in the formation of proteins? I told you DNA help in the formation of proteins and things like that. But how does it actually work? Let's see that. So over here, this is a nucleus, my dear children. The DNA cannot come out. But who makes the DNA? Who makes the protein is outside the nucleus. So what do? Basically, you can transcribe this information and form something else, and that something else is what? RNA. What RNA? mRNA, which you call as messenger RNA, my dear children. This messenger RNA is read with the help of tRNAs in the cytoplasm. And where this particular information is getting translated, I am able to understand what proteins have to be made. What do you call this process as? You call this as translation, where I am finally making the proteins. So you have DNA replication from DNA. So if you have DNA and if you want to make more DNA, you call this process as what? Replication. From the DNA, if you want to make mRNA, what do you call this process as? You call this as transcription. From the mRNA, you are going to plan to make proteins. What do you call this process as? You call this as translation. And this is the central dogma. And this is known as the central dogma, which was proposed by Francis Crick, my dear children. So how does the DNA help in the formation of proteins? This is how it helps in the formation of proteins. Cool? Chalo. Next. What does the transcription unit contain? Next question. So I said transcription will be beginning. So now from this point onwards, we are going to talk about this particular process, that is transcription. So what exactly, how exactly does transcription take place? So there is a unit in the case of transcription. Okay, this is the DNA strand, my dear children. Okay, I have divided this DNA strand into different segments. One is the promoter sequence, then you have the structural gene which is over here, then you have the terminator sequence over here. Okay, I have mentioned one strand as template strand, another one as coding strand. But ma'am, I don't understand. I am going to tell you. So a transcription unit in a DNA is defined primarily by the three regions of the DNA. If you are asked to make the transcription unit, or if you are asked to say which part, suppose this is A, suppose this is B, you are asked which is promoter, which is terminator, you should be knowing it. I'll tell you how. So this is promoter, terminator, and this structural gene, and you are going to transcribe the DNA. Basically, for transcription, you are making RNA, right, out of it. So for that you need RNA polymerase. You need DNA dependent RNA polymerase. Now RNA polymerase also works in 5' to 3' direction. So this particular strand that we see over here, this is the one that is going to get transcribed. What about this strand? This strand is not going to get transcribed. Why? Because if both the strands are going to transcribe, they are going to make pair. Again, complementary pair, that is a big problem. Apart from that, both of them will be coding for different proteins, and that will lead to a lot of confusion. We do not want that to happen. Okay, these are two important points. Now, suppose this strand is getting transcribed. I will call this strand as template strand, my dear children. Another one does not code for anything. I will call it what? Coding strand. Now you might think, why is it named like that, ma'am? It is actually named like that. I don't know why, but yes. So this is a coding strand which is not going to get transcribed. Template strand is the one that is going to get transcribed. Now if you are asked where is the promoter, you say at the 5' end, near to the 5' end of the coding strand. You do not give reference to the 3' end. Anyways, all the work has been done by 3', 5' template strand. So you have to give some work for the coding strand, otherwise it will feel sad. So basically you say the promoter is situated near 5' end of the coding strand. Similarly, the terminator is located towards the 3' end of the coding strand. Okay. Now apart from that, there will be additional regulatory sequence that might be further upstream, that is above it or below the promoter sequence as well. Now this whole thing that you see over here, it is the transcription unit. Cool. Now we go ahead. How does the transcription take place? Now that is kind of very much interesting. Again, you have initiation, elongation and termination. Here the RNA polymerase will bind to promoter. There it was original replication, right? Here it is going to bind to the promoter. And it says that I cannot go alone and bind to the promoter. I need a factor, and that factor is sigma factor. That factor is what? Sigma factor. With the help of it only, this guy will come and attach over here on the promoter sequence. Now the DNA helix will open and the transcription will slowly begin. Elongation happens. The RNA polymerase add nucleotide triphosphate. Following the complementary base pairing rule, only a short stretch of RNA remains attached to the enzyme. If you see, short stretch of RNA is only remain attached to the enzyme. Now comes time for termination. You are done with whole transcription. Okay. So RNA polymerase reaches the terminator region, my dear children, at the end, which is near the 3' end of the coding strand, right? There it needs another factor to terminate the process, and that is known as rho factor. What do you call it? You call this as rho factor, that helps in termination, causing the RNA to fall off. After this, the RNA will be separated and this guy will be separated. Okay, so RNA polymerase detaches, completing the process of transcription right over here. Got it? So this is how things work. Please remember sigma factor for initiation and rho factor for termination. If you take PQRS, R comes first, right? So you always remember R for initiation, rho factor, sorry. Oh my god. How do you remember? Okay, you can remember it like this. Okay, SRK, Shah Rukh Khan. Okay, so here S, that is sigma, R, that is rho factor, which comes later, and K, I don't know what is it. You can think for your own and let me know also. So but this is how you can remember. Sigma factor is the one that comes, attaches first, and it looks like this rho factor. It basically looks like a P. Okay, cool, done. Let's go ahead. Now, there's a difference between prokaryotic transcription and eukaryotic transcription. And you might ask, what is the difference? I'll tell you. Basically, we say, you know what, in the DNA or whatever is there, the segment in the DNA, suppose this is the whole DNA, the segment in the DNA that codes for a polypeptide, that is basically a protein or proteins, you call this segment as cistron. Okay, what do you call cistron? Now, in the case of prokaryotes, that is the one where there is no nuclear membrane, cell, or say, cell, membrane bound cell organelles and things like that, they have polycistronic DNA. What do you mean by that? So, over here there will be one structural gene, my dear children. You saw structural gene, right, between the promoter and terminator. There will be one structural gene that will be coding for multiple proteins. So, that means the genes, there will be many genes. There will be many genes that will be coding for multiple proteins. So, I won't say one structure, okay. So, one structural gene codes for multiple proteins as well. There are so many segments over here, so many segments each coding for each protein. What about eukaryotic transcription? They are known as monocistronic. Why? Because in their DNA, my dear children, suppose this is a cistron. This cistron consists just of one gene, and this one gene is going to code one protein only, nothing else. This one gene is going to code for one protein. Over here, there might be multiple genes coding for multiple proteins, which is why it is known as polycistronic. Okay. Now, prokaryotes do not have introns. What do you mean by that? Basically, if you talk about general thing, okay, if you talk about the DNA sequence, there might be some sequence, okay, like this, which are known as introns, which might not code for anything. They're just there. They are just intervening there. They are known as introns. And that is a huge problem. They have to be removed later on. Okay. So, in the DNA it's there. In the RNA which is formed from transcription, my dear children, that I forgot to tell you. So, I told you until now it's the mRNA which is being formed, It's not the mRNA that is being formed. The unprocessed version of RNA which is there, it is known as heterogeneous nuclear RNA, hnRNA, which is being formed. So, hnRNA will have all the things that was there in the DNA as well. Suppose DNA had the introns, then hnRNA will also have the introns. So, these introns have to be removed and exons have to be joined together, if introns are there. But in the case of prokaryotes, they do not have introns. What about eukaryotes? Eukaryotes definitely have introns as well as exons. Okay, will you be able to remember it? So, polycistronic as in their one structural gene will be coding for multiple proteins, as you might have seen in this thing, lac-operon. If you have studied lac-operon, okay, today we will be studying it. In lac-operon, there are multiple there's one structural gene, but these segments, there are different segments, okay, which will be coding for different proteins as well, beta galactosidase and this and that and everything. Okay, that is one thing. Now, they do not have introns, but we eukaryotes have introns. What about transcription and translation occurring? Do they occur in the same area or different area? So, basically, you know, there is no nucleus and everything in the prokaryotes. So, the DNA which is there, it is transcribed into mRNA directly in the same area, and that same area is cytosol, and the same area this mRNA will be making protein, that is translation will also be happening. So, transcription, translation, everything will be happening in the same area in the case of prokaryotes, whereas in the case of eukaryotes, my dear children, it will be occurring in different area. Transcription, that is formation of formation of mRNA from the DNA, will be occurring in the nucleus. Translation, this mRNA will be sneaking out of the nucleus, going into the cytosol for translation process. Works. So, these are few differences that you need to remember. Okay. Now, going ahead. RNA polymerase and RNA processing in eukaryotes. As you saw that eukaryotes, things are quite different. So, we have multiple RNA polymerases. We do not have single RNA polymerase. We have multiple RNA polymerase. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Do we have to remember them, ma'am? Yes. You have to remember what are they going to transcribe. So this is the one that is going to form the rRNA, my dear children. 28S, that is the unit, sedimentation coefficient rate, okay. So 28S, 18S, 5.8S, all type of things are being transcribed from RNA polymerase 1. What about RNA polymerase 2? They transcribe hnRNA, that is heterogeneous nuclear RNA. Okay. What about RNA polymerase 3? It is going to transcribe tRNA, my dear children. Okay, that is transfer RNA. Transfer RNA is the one, as I said, which picks the amino acids and everything. So you have to remember what does what. So for the transcription of hnRNA, that is the precursor of mRNA, I told you hnRNA is formed first, then processing is being done. So this thing has been done by RNA polymerase 2, most important. Now for rRNA, obviously, ribosome is the priority for RNAs. So rRNA which is there, it is formed first, that is ribosomal RNA. This comes and makes the ribosomes over here. Okay. RNA polymerase 3 transcribe tRNA, 5S rRNA and snRNAs. Remember the names. Now there is known as something that is known as RNA processing. What do you mean by that? Basically, the removal of introns which is there, you have to remove it, otherwise we would be having non-coding regions also. We don't want that. This process is known as splicing, which happens in us because we have introns, but not in prokaryotes because they do not have introns. Then there is capping, which basically adds methyl guanosine triphosphate at the 5' end. So if you see over here, there is 5-methyl, at the 5' end there is methyl guanosine triphosphate. Okay, methyl guanosine triphosphate which is being added up over here. This process is known as capping. This tells that where the mRNA has to go and things like that. Similarly, you have tailing. Tailing means at the 3' end, the 5' end capping was done. At the 3' end, my dear children, tailing is done where 200 to 300 adenylate residues are added at the 3' end. How much? 200 to 300. This is known as tailing. Now, until now it was known as hnRNA. But now this processed version of it will be known as mRNA, and this will be finally transported into the cytoplasm for the process of translation. Cool? Works? So that is about eukaryotes. So RNA polymerases, 3 of them you remember. 2. Second one is the one that makes hnRNA. Third one is the one that makes tRNA, and also 5sRNA, rRNA. Okay, 5sRNA, ribosomal RNA itself, but here it's just rRNA. 28S, 18S and 5.8S, and also small nuclear RNAs. Remember, I'm telling you again and again. Now, genetic code. So, how do they exactly, the mRNA, how do they exactly make the proteins? For that, I want you to understand this thing. Suppose this is an mRNA. This mRNA strand, my dear children, will be having codons. So, for example, AUG, UUU, UUU, or at the end UAA and things like that. Okay? This is an mRNA. On this mRNA, I am basically explaining translation also over here. Listen to me carefully. And this is the 5' end and this is the 3' end of the mRNA. Okay? You know, there is an organelle which is there, which is known as ribosome. So in between these two ribosomes, the mRNA is sandwiched. Okay? And the ribosome, there are certain segments where the tRNA can come and attach. Suppose this is the tRNA. And always the tRNA will be carrying one amino acid. I am saying that here you have certain nucleotides. Let me group them into triplets. Okay, let me group them into triplets. Now I am going to say right now that AUG is actually known as a codon, and if the tRNA opposite to it comes and binds to it, that means A will be having U over here, U will be binding with A, and G will be binding with C. How will it work? It's cool, right? This is known as anticodon loop of tRNA. It has the anticodon. Now, AUG will always bind with this, and this particular tRNA will always have a specific amino acid, my dear children, which is methionine. So that means this code is very specific for amino acid methionine. Similarly, another one will come having AAA, and this will be having another amino acid, that is phenylalanine. That means UUU will be always for phenylalanine. What do you understand from this? The mRNA, the codes which are there, they come together in the form of triplet to form codon, and each codon, my dear children, is going to code for an amino acid. When this amino acid comes together, what do you get at the end? You get protein. Now over here when it reaches, so all the this particular tRNA is being released after the amino acids are being done. Okay, so for example, this tRNA came, gave the amino acid. Similarly, another tRNA came. Now what will happen? Slowly, this thing will be going off. They two will be linked. Again, another will be coming and another one will be linked. So that's how the chain of amino acids are being formed. At the end, my dear children, when it reaches this, there won't be any tRNA that will be coming and attaching over here. That means this code, UAA, will not code for anything. What can I call this? I will call this stop codon. This will stop the process of translation to happen, and hence you got the protein. Okay, and AUG, I told that basically it is the one that codes for methionine. I am also telling you one more thing. This is also the one that is going to act like a start codon. This helps in the process of starting the process of translation. Got it. Now I am going to talk about genetic code. So over here you see. So you have these many genetic codes, my dear children, and if it comes to this direction, it would have been really great, but it's not coming. Okay. And if you see, UUU for phenylalanine, UUC for phenylalanine, UUA for leucine, UUG for leucine, and things like that. For all, we have 20 amino acids, and for 20 amino acids, we have codes, my dear children. So who was the person that basically said that there are triplets that are coming together and things like that? It was none other than George Gamow. He was a physicist. Okay, so according to him, he said that there are 4 nitrogen bases: adenine, guanine, cytosine, and uridine in the case of RNA. So 4 of them, if they come in 2 number, so it will just code for 16 codons. But how many amino acids do we have? We have 20 amino acids, so this is not enough. What about them coming in the group of 3? Then it will be 64. That's fine. It's about 20 anyways. There will be more codons, but it's okay. Probably there can be a case that multiple codons can be coding for the same amino acid. Yes, multiple codons are over here. They're coding for the same amino acid, my dear children. That is possible. So extra is fine. So he's the one who said that they come in triplet codon. Who? George Gamow. He was a physicist. Now let's see the features of the genetic code. So first salient feature is the codon is triplet, my dear children. Among them, 61 codons code for amino acids, and 3 codons are there that does not code for anything. They are acting as a stop codon. And what are those? UAA, UA, UAG, UAG, and UGA, UGA. So UA, UGA, and UAG. 3 of them, they are the terminator codons or stop codons. So this is one salient feature, this is another. Okay. Next thing, some amino acids are coded by more than 1 codon. Some amino acids which are there, they are going to get coded by more than 1 codon. That means, if you see, this, all of them they are coding for serine. That means more than one codon is there for coding for amino acid. So I will call this feature of codon as what? Degenerate feature. Next thing, if we see the codon is read in a contiguous fashion, there is no full stop, comma, anything. So that is there. Now the codon is nearly universal. That means, for example, from bacteria to human, all of them, my dear children, here the codon will be universal. UUU in bacteria, UUU in us, all of them will be coding for phenylalanine. Okay. Now one more thing. There is a difference. Okay, some exceptions to this rule have been found in mitochondrial codons as well as some protozoans. So in some protozoans and some mitochondrial codons, my dear children, you see something different over there. Now one more thing. AUG, which is there, okay, the first codon that I was talking about, it has dual functions. What are the dual functions? First, it codes for methionine, which is a amino acid. Another, it acts as an initiator codon or a start codon. So these are the features that you should be definitely knowing. So how can they ask you a question is that they can give you a strand and they will ask you what features of genetic code is being shown up over here. So if you see there are multiple codons coding for the same amino acid, you say that this is a degenerate feature. Okay, if you are saying there is no full stop, comma, it's a contiguous feature, whatever you want to say, but mostly it will be degenerate or things like that. Okay, so you should be able to spot it. So if you are not aware about all these small terms, my dear children, so if you are not aware of the small terms, my dear children, you will not be able to answer. So that's why I am telling you, please go through these things very properly. Okay. Now next thing. Now over here, one more thing. The discoveries were written in your book and you should be knowing about them. So there was an enzyme that is Severo-Ochoa, which was an enzyme. And what was the use of this enzyme? Basically, it synthesized RNA without needing the DNA. So usually we take DNA and on the basis of it we transcribe it into RNA, right? So with the help of this particular enzyme, the RNA was made without the help of the DNA. So we can make whatever basis we want, okay? But this is an enzyme. This is an enzyme that can be used for the process. Now no one was using it. Hershey-Chase Khurana came. He basically chemically synthesized the first RNA strand using this particular enzyme which is there. That is Severo-Ochoa, this polynucleotide phosphorylase. He made his own RNA. That means he knows that I have to put this particular base and this is the sequence of the bases which is there. Okay, this is the sequence of the bases because I am only creating it anyways. Now came Marshall Nirenberg. He developed a cell-free system, that is outside the living cell. He experimentally made the proteins. So basically out of the RNA he was able to make the proteins. And obviously this guy already knew the code which was there. And now the other guy made the proteins out of it. So obviously you know what code was there for what amino acid. So that's how all the codons were being made. Okay, there might be obviously other scientists also who work behind it, but this is the initial information which is there. So Severo-Ochoa, that is an enzyme that is what polynucleotide phosphorylase that is used for synthesizing RNA without the use of DNA. Hershey-Chase Khurana made it, and then Marshall Nirenberg, he basically developed a cell-free system, that is he made the first protein, not first protein, basically he experimentally made the protein outside the cell and experimentally confirmed which codons code for which amino acids, and that's how this particular thing was being made. Cool? And now we would be probably talking about, what do we have? Translation. Yes, we would be talking about translation, but I'll take a break and you also take a break and then come. Okay, please drink enough amount of water. Yeah? Okay, so let's go ahead. Over here, my dear children, there is a small thing given about mutation in your textbook where you might have seen this particular example, Ram has red cap. So basically, I told you that codons come in the form of triplets, right? So we have 3 codes. Now, what if one extra base comes in middle? What happens? So basically, first the codon was read like this. Ram is one thing, has is another thing, red is one thing, and cap is another thing. Now over here, if I include B, then the reading frame which is there, it is getting shifted. So it is Ram, has, Bree, and DCA, and then whatnot. So if you see, the reading frame is being disturbed, my dear children. In this case, obviously, the codon which is here will be giving rise to some other amino acid, and if there is some other amino acids, then obviously there will be some type of, let's say, mutant type of protein which is being produced over here. So these mutations are altogether known as frame shift mutations, or what do you call as deletion mutation, and obviously this would be leading or basically causing one severe mutation that can be either leading to some disease or can be disease free as well. So insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. This is known as frame shift mutation. Now there is one more thing that is known as point mutation, my dear children. What do you mean by that? Point mutation is basically when only one single base pair is being changed. For example, in the case of sickle cell anemia, okay, you had GAG codon which was coding for glutamic acid. Now instead of A, it was replaced by U, my dear children. Now this particular codon will be coding for something else, that is valine. Well, the amino acid which was there, it was a group of amino acids which was giving rise to what? The beta globin chain of hemoglobin. So if you have some mistake in the amino acid, obviously it is going to cause mutation up over here. If it is going to cause mutation up over here, that means the structure of hemoglobin is going to be disturbed. If the structure of hemoglobin is going to be disturbed, that means the red blood cell which is going to carry it will also have some changes in them. So which is why it becomes what? Sickle shaped. So this is the example of point mutation, my dear children. Now one more question to you guys. If I insert 2, it's okay. You're basically shifting the frame. What if I insert 3 things together? So Ram has, and I include B, A, C over here, and then is the same thing, that is RED and has. Got it? Yeah, sorry for that disturbance caused. Yeah, so I was saying basically when you insert a 3 base pair over here, insert 3 base pairs over here, you would be still producing the amino acid which was used to be produced, but again one amino acid can be extra over there, or probably if this is a stop codon, it will be stopping the process of translation to happen. So that is definitely a problem. Going ahead. Now we are going to talk about translation, my dear children, and for translation what do you need? You need transfer RNA, which is also known as soluble RNA. So it was used to be known as soluble RNA, that is sRNA, and this is the structure of it, where, my dear children, it's an RNA, so obviously it will be having a 5' end and a 3' end. The amino acids which are going to come over here, what's the role of tRNA over here, my dear children? If this is mRNA, the codon is there will be anticodon coming from the tRNA sites end. So tRNA will be having some anticodon, and attached to this particular tRNA there will be an amino acid. Now where does an amino acid come and bind? On the 3' end the amino acid will be coming and binding. So I will call this end as amino acid acceptor end. What do I call it? Amino acid acceptor end. Whereas over here, this loop which is there, what do you call this loop as? This is known as anticodon loop as well. Okay, and what do you call this process where the amino acid is coming and attaching over here? You call this process as charging of tRNA. You call this process as charging of tRNA, or you also call it as amino acylation reaction. Okay, both the terms can be used, and it uses lots of energy as well. There is a requirement of ATP up over here. Now there is a small thing given in your NCERT, that is the secondary structure of tRNA has been depicted that looks like a clover leaf. Okay, this is the secondary structure. Remember, the secondary structure looks like a clover leaf, but actually in actual structure the tRNA is a compact molecule which looks like an inverted L. Okay, inverted L, my dear children. Cool? Will you be able to remember this? Let's go ahead. Next thing. Now, what are the processes which are there and what all things are being involved? So I told you the tRNA charging is the first thing that happens, okay, charging of tRNA, where the amino acid are activated during using the ATP and are linked to their specific tRNA. This is known as amino acylation. Charged tRNAs facilitate peptide bond formation. So you know, amino acids, basically what is the use of it? Amino acids comes together, and this is what a peptide bond that is there. When amino acids, all of them, they come together, what do they form? They form proteins, my dear children. What do they form? They form proteins. So tRNA charging includes what? Basically that means amino acids are going to attach on the tRNA, my dear children. Okay, and this is happening in the first phase of translation. This is not the first step, this is happening in the first phase of translation. So tRNA, all of them are ready. Now what do we have? We have ribosomes over here. So these are the ribosomes that we have. We have a larger subunit and we have a smaller subunit. So the ribosome is a protein synthesizing factory, okay, composed of structural RNAs and proteins. So ribosome itself is made up of so many RNA. What do you call this RNA as? rRNA, ribosomal RNA. And also there will be proteins which makes up this ribosome. It exists as two subunits, large and small, in an inactive state. They are not active. The large subunit has two sites for amino acid binding. So suppose this is the largest subunit, it will be having two sites where basically the tRNA will be coming, and if the amino acids over here, they will be making a bond over here. Now, amino acids are joined by peptide bonds, as I already told you, and this requires energy. The 23S rRNA in bacteria act as ribozyme to catalyze the peptide bond formation. Obviously, you need enzymes over there, and here, in the case of a ribosome, basically they have their own rRNA that is going to act. I told you RNA has what? Catalytic activity as well. So, in the case of bacteria, my dear children, there is 23S rRNA which is going to act like an enzyme, and it is going to help in the formation of peptide bonds. Cool? Now, contains, okay, so over here if you see the mRNA, always remember, the mRNA which is going to get translated, before the 5' end and after the 3' end, there will be certain regions which are not going to get translated, and you call these regions as untranslated regions. What do you call it? Untranslated regions, my dear little children. Okay, they are not going to undergo the process of translation. Fine. And why are they important? That is the question that is being usually asked. They are important for the successful initiation or for the successful process of translation. If they are not there, probably translation would not be occurring properly. Cool. Now, if you are asked, what is the first step of translation? Okay, what is the first step of translation in which what happens? Let's see. Okay. First thing, the mRNA is there, no? It comes and joins with the smaller subunit. This is the first point, which is not being mentioned over here. So, this is a smaller subunit that we are having. Onto this, the mRNA will come and bind. Then comes the tRNA, and then comes what? Your larger subunit, my dear little children. Cool, and that's how the whole complex is being formed, and then the whole reaction takes place. So, initiation is the first step where the ribosome binds to the mRNA at the start codon recognized by the initiated tRNA. The first tRNA that comes, my dear children, which will be having the anticodon for AUG, that is initiated codon, that will come and attach on the start codon from which the translation would be happening. Now, elongation is where amino acid tRNA complexes bind to the codons in the mRNA. So, as you can see over here, each tRNA will be bringing an amino acid opposite to it and making a coming and binding over here. Later, if you see both of these things, they are going to make a peptide bond. Then this guy is going to move and you are going to form a string. Similarly, that goes on until, my dear children, you either see this. Okay, until the stop codons come, the translation process will be happening. So, ribosome moves codon by codon, adding amino acids sequentially, and in which direction that is moved? It moves from 5' to 3' direction. Always DNA polymerase, RNA polymerase, this everything moves from what, my dear children? From 5' to 3' direction. Now, termination also happens. That means the end of translation should also be happening. So, a release factor binds to the stop codon, freeing the polypeptide. What happens towards the end? There will be a release factor that will be coming in and binding to the stop codon. So, stop codon, for binding to the stop codon, you do not have any tRNA. tRNA does not come and bind to the stop codon. So, who comes and binds? It is a release factor which will be coming and binding and stopping the process of translation. Works? Chalo. Now, over here, a question. Gene regulation can be controlled at what levels in eukaryotes? As in, for example, I want to control the production of protein or whatever is there. How do I control in the case of eukaryotes, where we have nucleus, where we have membrane-bound cell organelles and things like that? First thing, transcriptional level. Formation of the primary transcript. I can stop it there. When the DNA is going to make the mRNA, at this level, I can basically regulate the protein formation, or basically gene regulation. I can make sure that transcription is not happening if I do not want, or if I want transcription to happen, I can regulate it over here. That is the first level. Second, processing level, as in, it's not directly forming mRNA, right? It is hnRNA, which then later forms mRNA. So, I can control the process by controlling the processing, that is splicing, tailing and everything, capping and everything can be controlled up over here. This is the second thing that can be done. Now, next thing, transport of mRNA from the nucleus to the cytoplasm. So, you know, in the case of eukaryotes, we have the nucleus, inside the nucleus we have the mRNA. This mRNA has to move out, then only the process of translation will be happening. So, over here this can be controlled, okay, transport of mRNA can be controlled, and then what? Translational level as well. So, once this mRNA is outside, the level of translation can also be controlled up there. So, these are the things that you should be keeping in mind for eukaryotes. On these many levels, gene regulation can be taking place in eukaryotes. What about prokaryotes? Do they have a nucleus? No. So, basically, whatever has to be done, it can be controlled at the rate, it can only control the rate of transcriptional initiation. That is, from the DNA, when it forms the RNA, my dear children, at this level, during the initiation only in prokaryotes, gene regulation can be controlled. Either the initiation or transcription can be stopped, or it can be started. And how do they do that? Basically, what happens is that, suppose this is a gene, and every gene, basically I told you, it has a promoter region and it has a terminator region. Right. So, in the case of prokaryotes, they also do have an operator region up over here. What do they have? Operator region up over here. What is the use of operator? Basically, I do not want this gene to be transcribed. Okay, this extra gene to be transcribed, I do not want that to happen. So, basically, I would be making some repressor protein, and this repressor protein will be going and binding on the operator. Now, even if RNA polymerase will try to do transcription, it won't be able to do this particular transcription. So, this is the role of operator, my dear children. Cool. And this happens in the case of prokaryotes. Now, what is the example of this? Do you know lac operon? You might have heard of lac operon, right? So, lac operon is a basic example of this particular thing. So, this is the gene for lac operon. If you see, you will be able to understand really easily. So, this is the gene from here to here. So, this is the promoter region. This is the operator region. ZYA, there are 3 genes up over here. I told you in the case of prokaryotes, they are polycistronic. That means one particular structural genes will be coding for different proteins. So, here this is going to code for a protein that is known as beta-galactosidase, an enzyme. Y is going to code for a protein that is permease enzyme, and A is going to code for another protein that is transacetylase enzyme, my dear children. Okay, so basically who will disallow the process of transcription over here? The operator. But where do they get the repressor protein from? So just beside it, my dear children, there will be another gene that is known as regulatory gene. What is it known as? It is known as regulatory gene, which is going to regulate the things. Suppose the regulatory gene is going to produce repressor protein. Okay, it is also a gene, it will also lead to a protein formation. Suppose it is giving a repressor protein. This repressor protein will go bind on the operator, and do you think the whole process of transcription can take place? No. So, this is lac operon. Okay, lac, why do I call it lac operon? So, basically what happens, lactose is the main guy over here. So, these enzymes which are there, okay, the first enzyme beta-galactosidase, as I told you, okay, it is responsible for the breakdown of lactose sugar. It is responsible for the breakdown of lactose sugar. Ma'am, I am not understanding. Listen to me carefully. So, basically, this is an E. coli. This is a prokaryote and this is an E. coli, a bacteria. Now, E. coli would be relying on lactose for energy. So, obviously, it has to take in the lactose and break it down. That's when it will be getting the energy. Okay. And I have all the setup for breaking it down. But what is happening? Whenever there is no lactose, I do not want this particular thing to produce all this thing. There is no use of it. So, I want to keep it off, and for that I use the operator. Okay. So, usually when lactose is there, my dear children, this beta-galactosidase will be acting on the lactose and will be breaking it down into what? Glucose and galactose. Sorry, glucose and fructose. Okay, glucose as well as fructose. No. Glucose and galactose, sorry. Glucose and galactose, okay. Oh my god, yeah. So, this beta-galactosidase will be breaking it down into glucose and galactose, and that's how it gets the energy. Okay, this glucose will further undergo all the reactions which is needed. Now, what is the use of permease? Permease is the one that will allow the permeability of the cell membrane to be in this way that it allows the entry of what? Lactose, which is there in the medium. What is the use of transacetylase? So, when these processes are happening, my dear children, there are higher chances there are some byproducts being formed and you have to neutralize it, you have to break it down. That is being done by transacetylase. Okay. So, that is the usual case. So, when lactose is not there, this operon is basically closed. Okay, this whole thing is basically closed. Now, suppose the lactose is there. At that particular point, what will happen? When the lactose comes, it acts as an inducer. It will act as what? Inducer, my dear children. So, this is inducer. It will go bind on the repressor protein. What will happen when it go bind on the repressor protein, my dear children? This repressor protein won't be able to go and bind over here. Hence, there is no one to stop anything over here. Transcription would be happening and all these enzymes will be forming. So, this is on condition. So, this was switch off condition and this is switch on condition. This will only happen when lactose is there. When lactose is there, that's when it happens. Now, over here. Yeah, so discovery, it was elucidated by Francois Jacob and Jacques Monod. Basically, these questions have come so many times, you should be knowing about the name of the people who were behind this. Okay, now the structural, it has a regulatory gene. I told you it has a regulatory gene which will regulate. Now, this can be positive protein and negative protein, both, my dear children. Over here, in the case of lac operon, it is basically producing a repressor protein. Now, structural gene is there where you have ZYA. Okay, Z will code for beta-galactosidase that breaks down lactose into glucose and galactose. Y will be coding for permease that helps lactose enter the cell, and A gene which is there, my dear children, it will be coding for transacetylase. Now, what is the function and regulation? Lactose acts as an inducer. So, in the absence of glucose, lactose enters the cell via the permease enzyme. Okay, now the repressor normally binds to the operator, blocking the transcription, but over here lactose, or basically another form of it, allolactose, will inactivate the repressor, allowing the RNA polymerase to transcribe the whole reaction. So what do I call this regulation type? I will call this negative regulation. I will call the whole thing as negative regulation. Why, my dear children? Because over here mostly it is being closed with the help of repressor. So there is a negative regulation happening over here, but in your NCERT only it is written that positive regulation is also there, but it is beyond the discussion. So let's not discuss it. Anyways, it might not be coming. So you should be knowing it's a negative regulation that is being shown via the repressor. Mostly it is in the switched off condition. Only when the inducer, that is lactose or allolactose, is there, that's when it gets turned on. Cool. Remember the name of Francis, Jacob and Jacques Monod. Okay, works. Now we have Human Genome Project. What do you mean by that? So what is their aim? These are the aims. So to identify approximately 20,000 to 25 genes in the human DNA. They thought it was there, okay? To determine the sequences, that is 3 billion chemical base pairs that make up the human DNA. Store this information in databases to improve the tools for data analysis, and also to check with the other sectors to address the ethical, legal and social issues that might arise from the project. These were the aims. Now, Human Genome Project was a 13-year-old project which was coordinated by US Department of Energy and also the National Institute of Health. So there are two things who were behind the Human Genome Project, which started 13 years ago. Now, have any other organisms been sequenced apart from the human beings? Yes. So many non-human model organisms such as bacteria, yeast, Caenorhabditis elegans, that is a free-living non-pathogenic nematode, that is Aschelminthes, Drosophila, the fruit fly, plant such as rice and Arabidopsis, and have also been sequenced in this particular thing. Yes, so basically I told about the Human Genome Project and everything, but how did they actually sequence the whole human genome? How was that possible? So basically, genome sequencing approaches, there are two main approaches. One is expressed sequence tags, that is identify only the genes which are going to express as RNA, my dear children, okay? And there is one more thing, that is basically you just do the whole genome sequencing, that is sequence the entire genome, both the coding as well as the non-coding regions which are there. So what exactly is being done? DNA is extracted from a cell, first thing. It is broken down into small fragments, my dear children, okay? And these fragments are cloned into host. You are going to put these fragments into host because obviously you need to amplify it. You want to make multiple copies, then only we would, if we are not able to understand one particular thing, we can focus on the next one, right? So next was cloning and amplification. So it helps produce multiple copies of the DNA fragments for easier sequencing. Commonly used vectors for this were, I told you, inside the vector this particular DNA was being put. So what were the commonly used vector? It was bacterial artificial chromosome, my dear children, or it could be yeast artificial chromosome which was being used in yeast. Now, the method which was being used. So sequencing was done using automated DNA sequencers based on Frederick Sanger's method. So Frederick Sanger's method was being used for sequencing all the amino acids over there, my dear children. And how were they assembled? Okay, we got the amino acids and everything that was there, what was it coding, what genes were there and everything. But how were they arranged back? Basically, the DNA fragments were sequenced and arranged using overlapping regions. So there might be regions, for example, if one particular gene is ending with A, okay, another gene, I took it something like this, UAC and then AAA. So I basically know that this might be, okay, this might be a continuation of this. So after this, I know it's CCA that might be coming ahead. So it's not just AAA, they might have seen for two certain things and then went ahead. But anyways, the overlapping regions were being found. And later, the whole chromosomes and everything were mapped. Each sequence was annotated and assigned to chromosomes. Chromosome number 1, which is there, it was the last to be sequenced, completed in May 2026, which was not very far, right? So in May 2006, my dear children, chromosome number 1 was completely sequenced. Cool. Next thing. The human genome, what were the findings of the Human Genome Project? So they were up here for some certain things. There were certain aims to find 20,000 to 25,000 genes, to determine sequences of 3 billion base pairs. Were they actually able to do that or not? Let's see. So the human genome contains 3164.7 million base pairs, which is very close. The average gene consists of 300 bases. That is a normal gene that you see. The average of them, most of them, they consisted of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin, that's a protein, with at 2.4 million bases. So this particular gene that codes for dystrophin has 2.4 million bases, my dear children. Now total number of genes is estimated 30,000, much lower than the previous estimates of 80,000 to 1,40,000. So total what my dear children, 30,000 genes were the ones that they were actually able to find out. Now the functions are known for over 50% of the discovered genes, less than 2% of the genome codes for protein. So only 50% of the whole genome we understood which of them are basically we were able to discover the genes, and they were only up to 50%, my dear children. The functions, okay, the functions of these genes, we were only able to understand only up to 50% of these genes. Now next thing, less than 2% of the genome codes for protein. Okay, less than 2% of the gene will be coding for proteins, other will be just non-coding regions, probably not so useful regions which are there. Now repeated sequence makes a very large portion of the human genome. So there are sequences which are repeated, which are not going to code for anything, but just there. Okay, repetitive sequences, stretches of DNA that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosomal structure, dynamics and evolution. So we can say, okay, in this particular organism, these many repetitions are found, but in us, these many repetitions. So basically, it gives a clue about evolution and everything which is there. Now, chromosome number 1, which was sequence, the last point, okay, it has most genes, that is 2968 genes, whereas chromosome number Y, that is found in males, it has the fewest, which is 231 genes. Now, scientists have identified 1.4 million locations where single base DNA differences are there. That means between you and me, there's a single base DNA differences at 1.4 million locations, which is why probably we are different because of all these differences. So these are the findings of Human Genome Project. Please remember certain very important things which I have underlined over here. Cool. Next thing, DNA fingerprinting, which is the last part of this particular chapter. So when is DNA fingerprinting used? So whenever there is a paternal maternal issue, we do not know who is the parent, or when there is a criminal case, there is a crime that happened, no one knows who did it. So basically at this point of time, something comes into play, and that is DNA fingerprinting. DNA fingerprinting works on certain logics, and we have to know about that. Let's see. So the total human genome is made up of 3 billion base pairs. So they are come in a set of 2. So basically you're talking about 6.6 in 10 to the power 9 million. Okay. Now over here, 99% of 9 of this DNA is identical between you and me, because that means only 0.1% difference is there. Okay, 0.1% of 3 million is basically 3 million base pairs are different between you and me. This means that, yeah, same thing. 3 million DNA bases vary between individuals. Now, DNA fingerprinting overview, how we can use this information. So, instead of sequencing all 3 billion base pairs to compare two people, scientists focused on specific DNA regions that are different. These variable regions, that is called repetitive DNA, contain small DNA sequences that repeat multiple times and vary between individuals. So between you and me, we would be having different repetitive DNA sequences. That is unique to us, just like our fingerprints. Just like our fingerprints. By analyzing these differences, scientists can identify individuals quickly. So, my dear children, okay, so with the help of these repetitive sequences, which is unique to me and you, we can actually find out who is the culprit or who did what and everything. There is something like satellite DNA and polymorphism. So our DNA has bulk DNA, the major part of it, and satellite DNA, small but unique repetitive sequence. So repetitive sequence are coming under the satellite DNA. Now satellite DNA varies from person to person and helps in DNA fingerprinting. Since these sequences do not code for proteins, mutations in them do not affect our health, which is why we are having different repetitive sequence. If probably it was something that was coding for protein or something like that, it might be we all might have had it. But as of now, it's not like that. So mutations might be happening, which is why we have changed repetitive sequence. So they accumulate over generations, making them useful for identification. Okay, so now we are using the repetitive sequence for DNA fingerprinting, and let's see how we are going to do that. So first thing, for example, I see that in one particular area some blood stain is there, and suppose this blood stain is not of the victim who is lying over there. This might be of the culprit. So we took the blood stain, we extracted the DNA out of it, Okay, we make a complementary DNA out of it. Okay, we made a complementary DNA out of it. So this is the strand that we are having. This is the strand that we had. We make a complementary strand, and now I'll call this as a probe. I'll call this as a probe. I'll mark it with some radioactive stuff. I'll mark it with some radioactive stuff. I'm going to tell you where I'm going to use it. Listen to me carefully. So now what is being done is that my DNA, suppose I have two people who have, I am a person who is the culprit, and there is one more person who is not the culprit, but still they are being sentenced. Okay, they are being checked whether they are or not. So our DNA has been isolated, my dear children. After that, the DNA has been cut using enzymes, and you call this restriction digestion. And then after that, the DNA has been separated using the electrophoresis chamber, okay, depending on the length and everything like that. Then blotting is being done. That means a paper is being taken, nitrocellulose paper is being taken, and whatever is there on the gel electrophoresis is being exactly put down on the paper as well. Now we have everything over here. This is known as blotting or southern blotting. Okay. Now what do? We do hybridization. Basically the probe which is there, I'll send the probe over here. I have two things. Okay. This is A, the other guy, and this is B, that is me. So I will be having a sequence like this. Okay. And now what I'm going to do is, I'm going to send my probe, and if my probe will go and attach on my sequences, my dear children, okay, first thing, this particular DNA isolated, that is also being checked whether they are also sedimenting like, say separating like this only. So I saw that this and this basically is separating the same thing. Anyways, let me check with the probe also. So I took the probe. If it goes and attaches on my DNA, that means it has found its complementary strand. That means what? I am the culprit, because I have this thing as a unique thing. No one has it. And this is the use of repetitive sequences in us. So autoradiography is done where with the unique, banding pattern is observed with under the UV radiation. This radioactive probe which is there, it will highlight up. And that's how the whole process is being done. So this is Alec Jeffrey who found out DNA fingerprinting method, and he used VNTR method, that is variable number of tandem repeats were being analyzed. What is it? Variable number of tandem repeats were being analyzed using this particular thing, and that's how I found out, and that's how we end up this whole chapter, my dear children. We started off from here where we spoke about the structure of the DNA. We went ahead with the nucleotide, nucleoside, everything. Okay, you have nucleosome, the histone proteins, the chromatin, euchromatin, heterochromatin, then the search of genetic material, Griffith's experiment, Avery and Hershey and Martha Chase experiment we did. Then we did a comparison between DNA and RNA. We spoke about which is the genetic material, what is the semi-conservative mode of DNA replication, and then we did another experiment to prove the semi-conservative mode of DNA replication. Okay, and then we spoke about the DNA replication. How does it happen? What is an enzyme which is there? What are the components which are needed for DNA replication? Okay, how does DNA help in the formation of proteins? We started talking about transcription. What is a transcription unit? What does a transcription unit contain? How are prokaryotic transcription different from eukaryotic transcription? We spoke about the RNA polymerases which are there in eukaryotes. We have different types. The processing, okay, splicing, capping, tailing, everything. Okay, then genetic code, we spoke about that. Who gave the genetic code? Who said that basically they come in triplets? It was George Gemau. Okay, and then Hargobind Khorana, Marshall Nirenberg, everyone came into the picture, my dear children. Then you had mutation over there. Then the tRNA structure, okay, tRNA charging and everything, translation process in a whole. And then the lac operon, the Human Genome Project, what was the use of it? What did they find? What is the use of DNA fingerprinting? And now we have totally completed the chapter. But still, if you feel that you have any doubt left, you have to ask me. I am available in the comment section or during any live that happens on Thursday. There will be a live for me, my dear children, so you can attend the live. I will be there to solve all your queries. Works? So that's all. Bye-bye. Take care, and do let me know if this particular video was helpful for you. I'll be so happy to see your comments. So bye-bye. Take care. Like, share and subscribe to our channel.